Thanks, Dan. This should be a lot of fun.
Yeah, this is going to be fun, all right. I'll be able to see myself on TV after this one. That's never happened to me before in all my years of presenting, so that's going to be fun. Welcome, thanks for coming. And yes, I was a biology teacher, and now I'm just a hobbyist, I guess, at it. And so tonight, as we look at some of the things God created, I just hope you'll be amazed. I won't say a lot about evolution, believe it or not, tonight, because what I'm going to show you is what God made. Because there is no other way to explain how these things came into existence. There just is no other way. So without further ado, let me just take you, first of all, here's your Midwest Creation Fellowship, some of the people involved here. You saw Ken speaking and Walt in the back. And do have a little commercial here for next summer. If you're interested, there is the International Conference on Creationism coming up in August next year. If you might want to participate in that, it certainly will be open to the public. And there is a website, icc.com, or creationicc.org. You can go there too and participate in that. I've been to 5 of them in the past, and Boyd and I, we went to the last one, wasn't it, in 2014? Yeah, we did. So that's coming up. A little bit about my philosophy in education. It's not my philosophy, it is the Word of God. Right here in 2 Timothy 2:24 to 26, it tells us what we ought to be like as teachers. Doesn't matter. Public school classroom, Sunday school classroom, and the Lord's servant must not be quarrelsome, but be kind to everyone, able to teach, patiently enduring evil, correcting opponents with gentleness. God may perhaps grant them repentance, leading to a knowledge of the truth, and they may come to their senses and escape from the snare of the devil after being captured by him to do his will. So that's my way of teaching. I'm not going to argue with anybody tonight and quarrel over anything. I really don't expect to. I am a teacher, though, and so my presentations, and even in the classroom, were never stand-up dialogues. They were always interactive with my classmates, the kids in the room. I engaged them in questioning, and I would come back to them with answers or probe their brains. So if I move into that mode, because here come a few students right here, I can tell. They're the ones that always bring backpacks. And I'm probably going to end up doing that. I'll default right into my teaching mode, and I'll be asking you questions. In fact, I've got a few in here already, I know that. Yes, I have 2 birthdays, so that you know about my spiritual life. I was physically born on January 17th, 1950, and I was spiritually reborn on January 25th, 1970. John 3:3 is a part of that narrative where Jesus talks to Nicodemus and makes the very unusual statement, at least to Nicodemus, about you must be born again. John 3:3. Well, that happened to me on that date. 2 Corinthians 5:17, anybody know it? That's exactly right. So then anyone who is in Christ, therefore, if any man is in Christ, he is a new creation. The old passes away, the new has come. A great verse. So that's about my spiritual life. Now another little plug, a commercial. It's the holiday season. It is the Christmas season here. And there is a book that I'm reading right now that I'm going to recommend to anyone interested in science-y type reading. This book, The Great Christ Comet, I'll give you a little bit more in detail, but talks about the Magi in this book and what did they see and what did they and why did they do what they did. We know the story. We all know the story, at least from Scripture. A verse of Scripture in Numbers mentions the event in a prophecy. A star will come out of Jacob, a scepter will rise out of Israel. And very interesting reading. I'll talk about the author in just a second as I come up on that. But just imagine this. It's been over a month of camel travel. You know a king has recently been born to the Jewish nation. You've been to Jerusalem, and no one else seems to know what has happened. As counseled, you press on, now southward toward a small village called Bethlehem. But where in Bethlehem? It's dark, no street lights, no lighting at all. You keep an eye on the moving star. It's straight ahead of you now. You see the town on the horizon, but which house? Down a shallow ravine and now up the other side. A star on the ravine horizon, you're keeping your eye on it. You ascend to the top and, gee, I wonder which house. It could have been that obvious. And the author of the book, Dr. Colin Nicol, who has his PhD from Cambridge University in theology, and then decided to study astronomy. And the book goes into, it now will become the seminal book on this event, this star of the Magi. But I'd recommend, if you're interested this kind of a thing, read the book. It's really fascinating, and it really could have been just like that. Okay, take a little look at this one here as I begin my presentation. We're going from small to large, bottom to top. If you read that center column, starting with energy, and we get subatomic particles, and from those we build the atomic particles, the protons, as mentioned earlier, neutrons, electrons. So with those we build atoms. Atoms we build molecules. Molecules we build bigger molecules, and with bigger molecules we build even bigger molecules. Those bigger molecules, if we think of living systems, become organelles like nuclei and endoplasmic reticulum and vesicles and all of those. Those are put together into cells, cells put together to make tissues, tissues, organs, organ system, blah, blah, all the way up. Organisms become populations, populations become communities, communities become biosystem. The bigger we're getting, now we include the entire biosphere. Add the atmosphere, the lithosphere, the hydrosphere. You've got the planet. Then you've got more planets. You got solar systems and galaxies, and then we have the entire universe. Got the idea? That's the progression. On the left side, you can see what we need to see these things. As things get so far out there in the universe, optical telescopes and radio telescopes help to bring into vision for our eye to see things that are way out there. And then as we go the other direction down on the left, we need microscopes and even more powerful microscopes, electron microscopes, to get down to see these little bitty molecules, because we're standing with right about in the middle there and this entire system. On the right, I have some units, some of which I'll talk about today. And then, so most of my discussion today is going to center around this area. This is what I taught, for the most part. This is biology within this size, including the ecosystems and biosystems above. Things that we could see for the most part. Of course, you remember studying cells and needing a microscope in biology class, remember, to get down and see those little things, and even smaller. Well, today we're going to go into this realm. This is where we're going to be. We're going to be discussing some of the biomolecules that make up these systems and cells. In particular, we're going to look at proteins. Now, how many of you were here the last time I spoke on proteins? Which was probably the last time I spoke, because I spoke on CRISPR. You'll remember CRISPR and that editing system, and before that I spoke on ATP synthase, which is another protein complex, when today I'm going to talk about motor proteins. So we're going to be in an area that we've been to before. God's world of proteins again, and I have a couple of animations here. Nowadays, this kind of technology and doing this kind of thing, animating these molecules so that biochemists and cytologists can better understand what we're looking at. Three-dimensional animation is very much a part of this science, and there's a lot of room for those who are interested in computers and animating in the world of biochemistry, believe it or not. Animating these processes is even more, the processes are even more difficult. I'll show you some of that today. I mean, it's this kind of a structure that I'm talking about. And highly complex numbers that are staggering in size and scales that are so small you can't even imagine. And yet there are things down there and very interesting things. Now, quick review here. Here we go now. I'm the teacher. Proteins are very large molecules made out of smaller molecules called? That's correct. Amino acids. All right? You can go to the health food store right now and buy a bottle of them if you'd like. You can simply get the amino acids, and in fact, you can go to a butcher shop or a local deli and pick it up in bulk as proteins, okay? But proteins are made of the smaller little molecules called amino acids. You could save yourself some digestive work by buying the right-hand side product there. What is that called? Hyperamino. Okay, anyway. There are only how many kinds of amino acids? 20 is the correct answer. Who was that? That's right. There are only 20 kinds of amino acids, and you have to imagine this is just like letters and a language. We only work with how many letters in the English language? 26. It's only ever been, as far as I can remember, 26 letters, all right? With those 26 letters, though, we can make a lot of different kinds of words. And I looked it up. How many words are in the Oxford English Dictionary? 176,000 plus usable words, not counting names given to biomolecules and strange 176,000. But when it comes to amino acids, it's going to be the assemblage of amino acids also to make various kinds of proteins, just like different letters make different kinds of words. Okay, now, show the beads for illustration purposes. Well, have the kids assemble the protein. On the floor up here, I don't know how many of you have seen it or have not seen it, is a string of beads. Each bead represents an amino acid. And what I had my grandchildren do, they're right here, raise your hand, grandchildren, the three of them, was to build the CRISPR-Cas9 protein. Based upon what we know about that protein, the sequence of amino acids, I took that sequence of amino acids and I converted it to beads. I converted each amino acid to a bead. I have a collection of 20 different kinds of beads here. Each was a different one of the 20 amino acids. And what they simply had to do, because I looked up the sequence of the amino acids, you could do this too, you know. It's online. There's several different sources of where you can find the sequence for any protein that they've so far sequenced, okay? So each of the kids got a piece of paper and had to start getting the right amino acid and putting it in the right order, which they proceeded to do here, as you can see, eventually culminating in a sequence of beads that is analogous to the sequence of amino acids that makes up the protein Cas9. 1,263 amino acids makes up one Cas9 protein. 1,263 amino acids make up one Cas protein. Each amino acid must be in the right order in order for this molecule to work. And they did it. It's laying right here. You can take a look at it a little later on. Here were the beads we used. Here's the collection that I have of beads, and each bead represents a certain amino acid. I have them all labeled there. Here are two what we would call my proteins made out of beads. Okay, not the ones the kids made, but those are two. Now, what happens? Here's a collection of my beads that represents a protein so that you know if you digest this, in order to digest this, you introduce it into your stomach where there's a pretty high concentration of hydrochloric acid. The pH is fairly low in your stomach, and that has the effect of taking a protein that is in this bundled up quaternary level of structure and reducing it to a string. That's what the acid does. It begins to denature the molecule so that it doesn't come apart, it unravels and becomes a string. Further digestion includes digestive enzymes, which will take that string of amino acids and disassemble them into individual amino acids. From there on, it's a matter of your bloodstream going through the small intestine. These individual amino acids are absorbed out of the small intestine environment, put into the bloodstream, and distributed to your cells, where your cells now will have a readily available supply of amino acids so that your cells can build the proteins you need. This is simply what you do to a hamburger. Upper left, product in your small intestine lower right, and those amino acids are distributed so that you can build the proteins you need to keep your body running and your cells need. Any questions on that? Yeah, go ahead.
How does your body know which protein to produce?
Oh, that's a good question. Yeah, how do your cells know what proteins you need? You're probably running on in the neighborhood of 200,000 different proteins in your body. Let's say you're running, I want to pick on a protein, let's say hemoglobin. You cut yourself, you're losing blood. The body can detect a drop in the number of red blood cells, and this does stimulate the blood marrow to begin to produce more red blood cells. Now, how is that sensed and communicated? I don't know, honestly, in that case. Lines of communication, though, and how communication occurs in this cell is a very interesting topic. And it is one that is more and more, I know this, being tested in AP courses and for essay questions of these different communications methods in the cell. Some have to do with concentration gradient variations. There are signaling mechanisms from cell to cell. There are feedback loops for all of this. It's a very complicated process. It makes a very already complicated system even more complicated. Now, what does that mean for us? It means an appreciation for what we are looking at. It means a questioning of the current dogma about this all being the result of chance, because the levels of complexity multiply on top of each other. It's not just complex, it's complex on top of complex, and complex things controlling complex things, making it even more complex. And then someone trying to tell me it happened by chance, I can't go there. All right, at any rate, communications is a topic. Now, when it comes to building your proteins and doing something with those amino acids, you understand that you inherited DNA from your parents. And what you inherited in your DNA is the instructions for how to take those amino acids and put them in the right order to make the right protein. It can't just be done. It has to which amino acid to put in order to make the insulin protein molecule you need or the hemoglobin protein molecule you need. And so where are the instructions to take all these amino acids that you're putting in your system and picking out the ones you need, the right one, and getting it in the right order? It's in your DNA. That's where the code is, on which amino acid to put in which order for which protein. And so, speaking of DNA here, and I have a molecule of DNA represented, just so you understand, approximately 2 nanometers wide, this molecule being represented at the top. Obviously, you see just a fragment of a DNA molecule on the left, displayed here on the left. I estimated in doing these calculations today that my image on the screen would be a foot wide. And I think it's a little bigger than that. If you could just imagine that image being a foot wide, let's say it's a foot wide, then all my calculations will make sense for you. If it's 2 nanometers normally, and we now blew it up to a foot wide, the question is, how long would it be? Would this diagram would be how long if I expanded this to 1 foot? Well, going to 1 foot is increasing the size of the molecule 140 million times. I did the work on that. It's 140. So this is 140 million times wider than it normally would be in a cell. The average length of a DNA molecule in a cell is 2 inches. That is, if you went to a cell and you got into the nucleus and you had a very fine little probe and you could lift up a single strand of DNA out of the nucleus, 2 nanometers wide, and you were to pull it out, on average, it'd be about 2 inches long. okay? You inherited 46 of those molecules, 23 from your mother, 23 from your father. So that each cell has 46 DNA molecules on. Those molecules are genes that code for the instructions on how to build the various proteins you need to operate the cell. Any question on that? Each one? Okay, 2 inches is the average length. Now what I have pictured at the top is a thing done in the hospitals. If they had a question about the number of chromosomes that an individual has, this because there are people born with abnormal chromosome number. Most really don't survive, but a few do. Suppose they have 46. So let's say there's a question about whether or not the baby being carried has the correct number of chromosomes. They simply do, in the upper left, they'll take a sample of cells from the baby, they take it to a lab, they put it under a microscope, they have a lab tech who looks through the cells for one that's in the stage of cell division, that is in a mitotic condition, where the chromosomes have been coiled up and bundled up into nice little packages and are actually visible. Normally the DNA molecules all spread out, you can't distinguish them, but when they're coiled up, super coiled up, you see them, like those represented in the upper left there. They take a picture of it, an 8x10 glossy picture, and hand this picture over to a lab technician who's simply got a very sophisticated tool called a pair of scissors. That technician simply cuts out every one of those little chromosomes and begins to pair them up, because you got to have 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s, all the way up to 2 number 23s. And if you got the complete set in the center like that, you're chromosomally normal. Doesn't mean you don't have mutations, okay? It just means you got all your chromosomes. In those chromosomes are the genes, and genes could still have mistakes, but at least you got all your chromosomes. This person does, and by the way, you also can tell they're a male. That's correct, very good. That person in the center is a male. Now, the DNA is all coiled up, nice and neat in those little logs that you see. What I have as a picture on the right is one of those logs that has been exploded. They removed all of the histones, which are the proteins that are spools on which they coil up the DNA, and they chemically dissolve those. And as a result, the DNA molecule just fell all apart. And you're looking at one strand of DNA, one molecule, but you could never follow those lines. That's one DNA molecule that's been disorganized. Let's put it that way. That's one. It would be that. So now, okay, it's 2 inches long, let's say. What is that? That's 50 million nanometers long. The average DNA molecule is 50 million nanometers long. That's the same as 280 million inches. Now, that would be at this scale, if we went up 140 million times, that would represent this molecule having to have to be 4,300 miles long in order to suit this scale that I have this width at. Does that make sense to you? Now you know why they cut it off here and they cut it off there. Because if they tried to show you the whole thing, it'd be 4,000 miles long plus. Yeah.
23 chromosomes, they each average 2 inches at this
Yes.
unraveled state. So is that where they get the 46 average length of the full genome?
Average length of the full genome?
Yeah, the full DNA molecule, all of the chromosomes.
Well, remember, you're working with 46 DNA molecules.
But aren't they in pairs?
They are in pairs. 23, 2 number 1s, 2 number 2s, 23 pairs.
So it's 23 times whatever length they are.
Okay,
Side by side.
oh, the 1 inch, the 2 inches
Yeah.
represents one of these.
One of 23 or one of 24?
One of the 23.
Yeah.
The average of the 23, because some would be 1/4 of an inch, some would be 4 inches, 2 inches would be the average. Okay? That was just rounding.
You get the total length.
Total length, you would have to add, okay, the total length of DNA, if you stacked them end on end, of the ones you inherited, this is never exists, but you'd simply take 2 inches times 48, or 46, what's 2 times 92?
But I thought you said that they were 23 pairs, so
Yeah.
if I keep the pairs
Times
together,
23.
Okay.
it's really 2 inches times 23,
Yeah,
because of the double helix structure.
yeah.
I'm just trying to get it straight.
Yeah, I know, okay. If you took, by the way, okay, in that vein, if you took all of these 46 DNA molecules, maybe this is where you're going, and you stacked them end to end so that you could get the total length of the DNA, now this never happens. You never have your DNA molecules all stacked end to end. There are 46 distinct DNA molecules in every nucleus, okay? But if you want to know the total length of the total amount of DNA you have, it's about 3 feet long, coming out of one nucleus.
Which is,
Well, that would be all of these uncoiled and stacked, all 46 stacked end to end. That would be about 3 feet. Okay?
trying to get to the math.
23 by 2, that would be 46 inches, so just short of.
Okay, I don't
You
ex-
don't get 3 feet out of that math then.
Well, okay, it's, first of all,
That's fine, so just keep going.
yeah, okay. Point being, if this represented one of your chromosomes, just one of the 46, this diagram, and right there it's a foot wide, we blew it up 140 million times. Its length being 140 million times now longer would be 4,300 miles long at that size. And that's one molecule. That's all one molecule. These molecules are huge and very complex. All right, I didn't really mean to spend a lot of time on this point here. It was just by way of, here are some other animations of some of the various proteins that you'll see in a cell that have been analyzed and animated. Here's where we're getting to where we want to be today, the motor proteins. 3 proteins exist in all cells, there may be more, which have a specific moving function. The function is to move something, and thus they're called motor proteins. The main ones are myosin, kinesin, and dynein. They look like this. They have this rather odd structure, and I just put this up there so that you can see. There are cargo-binding tail domains on one end, and the other end are called head ends, but that's sort of a misnomer. You'll see in a little while why that is. The action of these proteins is something like this. Let's see if I can get this to move. Okay, I'll go. Oh yeah, it was going, wasn't it? Let me back up to it. Oh, there it goes. Okay, it's one of those GIFs. This protein, in interaction with ATP, when it interacts with that molecule, makes a structural change, a change in shape. And this is how it looks. Let's say right now ATP interacts and the molecule changes to that shape. It lets go of the ATP and returns to this shape. Interacts, lets go. And so the overall structural change can be displayed actually as a moving, in this case, kind of pendulum. Now with that movement, we can do work. That movement actually can be tapped off to do work in the cell. That's how a motor protein functions. This is another one called dynein. I'll show you, so if I get this one to activate too. There we go. Same thing, dynein, you see we get this swinging shape in the interaction with ATP causes a joint, as it were, to change position, which causes this whole lower part to change in a gross way in this swinging fashion. Again, with these motor proteins, that can be tapped off to do work.
So you're half a figure student, what's ATP?
Okay, ATP is to your cells what gasoline is to your car. It is the fuel that runs all the biochemistry in your cells. Now, you don't eat food and get ATP. That is not what happens. You eat food so that your cells can make the ATP to use the ATP in these chemical reactions. ATP recycles in your cells, and you need carbohydrates, for the most part, to regenerate ATP. Okay? Now, here is our kinesin, right here. Of those 3 motor proteins, here's a picture of kinesin himself, actually caught in the middle of work. Now, when you see him working, that's going to be the funny part. All right? On the right is the sequence of amino acids which must be strung together to make one kinesin protein. This, in fact, is what's called a dimer. It is 2 kinesin proteins twined together. So you inherited a gene from your parents, at least one, for how to get the right order of amino acids together, 963 of them in the right order, such that they would take the shape of sort of a golf club driver. Okay? And you take 2 of those, and you make 2 of those, and you sort of twist them together, and now you have one kinesin protein. Okay? The swing action is going to be here, in interaction with ATP. Here's some other pictures in different positions of kinesin. Kinesin there on the right as a motor protein is going to move some cargo from point A to point B in the cell. The other bit of information that's important is that he must walk upon what is called a microtubule. A microtubule is to kinesin as a railroad rail is to a train. Kinesin must walk on microtubules. Now, microtubules themselves are proteins. They're made out of tubulin. That one's not hard to remember. Okay. And I think I have a little action displayed here, kind of what happens. You can see that the club head ends, because of structural changes in the protein, change position along the tubulin molecule down there in the lower right. Okay. A different animation, here's an actual electron micrograph of a kinesin molecule laying somewhere on the surface, and that has to be a very powerful magnification here to get that much. A little more detail displayed here. There's a tail side and a head side. There it shows the interaction of the heads interacting with ATP, causing structural changes in the kinesin molecule, and it then able to change its heads to different locations on the microtubule. Now, microtubules, a word about microtubules. In the cell, these tubules have twofold purpose. One, they give some integrity or support to the cell. Sometimes call that a cytoskeleton. Okay? Secondly, they actually act as highways within the cell. So I had the London Bridge here because this bridge does 2 things. It supports some residences, I think, there, as well as conducts traffic on the horizontal plane. Microtubules assemble and disassemble, and you talk about communication. These tubules can assemble in front of the kinesin as he's moving and disassemble behind him. How they know where they're going, one, I don't know as they know. How they know when to assemble and when to disassemble, I don't know either. But there are smart people out there who maybe know. But it is communication and cell communication. More on this in a minute. Here again are some close-ups of this track that they use for transporting. Tubulins come in They're also a dimer. There's an alpha polypeptide and a beta polypeptide, which together form the dimer tubulin. And these stack on top of each other, as you see there, in these kind of arrays and form a tube. And again, that can be a highway and also a strengthening segment for the cell itself. Here's another picture of kinesin in relation to the microtubule. Now, motion. I took this micro clip because things do move in a cell, and I know you've seen these before. What are we watching? A cell. Cell where? When? In mitosis. And those are the DNA molecules that are in those log forms. They're in the supercoiled condensed form because they are being moved. They've been replicated, and it's time for the copies to separate, move away from each other so that the cell can divide down the center, and we now have two new cells, each with an equal amount of DNA and information. You see what just happened. What needs to be explained, now here is another, let me halt this one just a second. Whoops, I'll come up to it again. Using some dyes and some different lighting effects, what we're seeing here now is into these same cells, but seeing close up those microtubules. You can see they fan out, and there's an array of them in the cell that is distinguishable. The cells are in various stages of cell division, as you can see here, but the microtubules are very much associated with cell division because the chromosomes have to be moved. They have to be pulled apart and moved away from each other, the copies, and that's going to involve motor proteins to do this job. See if I can get that rolling again. Here we go. Okay. A close look at what's going on. Our microtubules are connected to the chromosomes by means of a kinetochore, as you can see it there. And what's going to happen is that what we have is a DNA molecule on the left and its copy on the right. The two still remaining together at this state. We call these sister chromatids. And, but at this point, they're going to need to be pulled apart if we're going to get two new cells. Each cell must get a copy. So here's a little animation to kind of show how this lines up, and you can see the microtubules, a part of it. We're going to go in very close to that region in red, and we're going to see the beginning of the involvement of some of these motor proteins in this separation. The red area is called a kinetochore. This is the supercoiled DNA molecule. Here we see the microtubules getting larger and extending as construction on the ends of connecting tubulins in there is occurring. By the way, this is not real photography, you understand. This is all animation. What is occurring is far too small for a camera to photograph. So this is what would be called the best explanation for what occurs. They've never seen it happen, but with all of the chemical analyses and the laboratory experimentation done on this process, this is the best way to explain what happens at this moment. Now I'm going to leave it here and come back to this in a little bit, because we want to talk a little bit more, well, not right there. Here's kinesin and a cargo, and there's a microtubule. Another diagram of the same thing, kinesin with the heads down on the microtubule, and it connects at the other end with some kind of a vesicle. Now, in the research that originally investigated this worked with neurons. Neurons, as a cell, connect to other neurons, but not physically. There is a gap from one nerve to another nerve. They don't touch each other. There is actually a gap in there called a synapse. It's a synaptic gap. No nerve directly touches. There's a synapse. So when an impulse comes down a neuron and it has to move to the next neuron. When it comes to the gap, the impulse has to stop. But the question is, how does the impulse begin again at the next neuron so that it continues down? Neurotransmitters are chemicals that are sprayed into this gap. They move across the gap, chemicals, make contact with receptors on the other side. When enough contact is made and action potential is stimulated on the next neuron, boom, it moves to the end of that neuron, and there again it'll encounter another gap. Neurotransmitters are sprayed into the gap, and on that goes. It's in those gaps, by the way, where drugs have their effects, where they can identify why addictions occur and so forth. And I'm not going to go into detail on all that right now. But what they noticed happening was that at that point where neurotransmitters are supposed to be sprayed into the gap, there must be a source of neurotransmitters in the cell to come to the edge of the cell and spray their contents in there. And what's happening is kinesin drags bundles of neurotransmitters made somewhere else in the cell and drags them to the edge of the cell at the synaptic gap, and it's that cargo vesicle which will meld with the cell membrane and dump the contents into that synapse. So, well, this happens quite a bit. Neurons are firing all the time. You need a lot of neurotransmitter, so there's this constant cargo hauling by kinesins to bring these vesicles to those sites. That's how it was discovered.
The motor protein
Here
kinesin is a dimer with two identical motor heads. Each head consists of a catalytic core and a neck linker. In the cell, kinesins pull organelles along microtubule tracks. The organelle attaches to the other end of the long coiled coil that holds the two motor heads together. The organelle is not shown here. In solution, both kinesin heads contain tightly bound ADP and move randomly, driven by Brownian motion. When one of the two kinesin heads encounters a microtubule, it binds tightly. Microtubule binding causes ADP to be released from the attached head. ATP then rapidly enters the empty nucleotide binding site. This nucleotide exchange triggers the neck linker to zipper onto the catalytic core. This action throws the second head forward and brings it near the next binding site on the microtubule. The attached trailing head hydrolyzes the ATP and releases phosphate. As the neck linker unzippers from the trailing head, the leading head exchanges its nucleotide and zippers its neck linker onto the catalytic core, and the cycle repeats. In this way, kinesin dimers move processively, step by step, along the microtubule.
Get it? Okay, are you ready now? Here we go. Can you believe that? That's kinesin at work. Well,
It has to move a lot faster than that if it's moving vesicles or
well, yeah,
the release.
I've got that worked out too. Is this the craziest thing you've ever seen? I mean, okay, this is where it's funny. Okay, that's funny. This thing is just a molecule. There's no life there. It's just a molecule. All right? No brains. No. It's just changing shape. Again and again, the shape change initiates this flinging of the feet forward, and it's repeated over and over and over again while this guy creeps along a microtubule, lugging his load as he goes. Now the microtubule, think about this, talk about communications again. How does the microtubule know in what direction to make itself so that when kinesin's walking on it, he's getting his vesicle to where it's supposed to be? I don't know.
Amazing coincidence.
This is what I want you to see. I mean, it is fantastic. And okay. Here's another better animation, okay, of the process.
Cells are full of specialized components that perform functions vital to their existence. But how do these components get to the right locations inside the cell to perform their functions? For larger components, a transportation system is needed. Meet the kinesin. Masterpieces of microengineering, kinesins are miniature motorized machines that carry cargo
Notice the
from one part of
microtubule
the cell
assembling?
to another, walking along self-assembling highways called microtubules. Known as the workhorses of the cell, kinesins have 2 feet, or globular heads, that literally walk 1 foot over another along the microtubule, pulling their cargo to its destination. Each foot possesses 2 special locations called binding sites that interact with other molecules. One site attaches to the microtubule and the other binds with ATP, the energy molecule of the cell. When one foot binds with ATP and uses its energy, the foot flips over, resulting in the walking motion. Each foot has a short neck which is connected to a strand of a long coiled stalk. At the end of the stalk is a fan-shaped tail which holds tightly to the cargo being transported. Kinesins can carry cargo that are many times their own size. Sometimes a kinesin is in danger of getting stuck on the microtubule highway because of blockages caused by other cellular components. To get around such obstacles, multiple motor proteins may be used to carry a single piece of cargo, together providing enough force to break free. Kinesins typically walk away from the center of the cell and toward the cell's periphery. The kinesin's 2 feet work together efficiently, with 1 foot holding fast to the microtubule, while the other releases itself and takes a step forward. This coordinated stepwise movement allows kinesin motors to walk as many as 100 steps per second, moving about 8 nanometers with each step. When not carrying cargo, kinesins can shift to energy-saving mode to conserve fuel until their next job. The kinesin plays a vital role in many cellular processes, not just transporting materials, but also aiding cell replication. The walking kinesin molecular machine, another example of intelligent design.
I'll bring you back to that kinetochore here, and again, the job of separating 2 sister chromatids. As he mentioned, kinesins always carry their cargo from deep within the cell outward, that direction. Dynein, the other motor protein, always carries cargo from the outer periphery toward the center, along microtubules. Now, as the moment for separation occurs, there's a change in color here by the artist who's doing this, but there's going to begin a migration. Here it is. Dynins begin to move down the spindle fibers. This is going to trigger a pull, ultimately, on the... now, yeah, you can see there's a pair of kinesin. Don't ask me, I don't know why they're coordinating that way, but anyway, there goes a dynein right there. Looks like he's dragging a kinesin against his will, maybe. I don't know.
Wow.
Oh, yeah.
Am I the only one where they are moving sideways, actually?
Well,
Kinesin.
moving sideways?
Yeah, it's like the side.
Oh,
It's not-
like a karaoke style?
Yeah,
Okay.
something like that.
Well,
I mean, if you look closer, you'll see that the
Yeah.
feet are basically the same, you know.
going like that, yeah, yeah. Well, it's definitely not our style of walking. What we saw here in just a, yeah. Okay, so the combined effect of all of those motor proteins moving on the microtubule can exert a force on the sister chromatids such that the centromeres begin to break. And the copies of DNA then are pulled, literally dragged, to opposite ends along those microtubules. And now we have two cells, the DNA molecules uncoil out of their log form and into their chromatin form, and you see two distinct nuclei in the end. So 800 nanometers per second, well, that's equivalent to 2 millimeters per hour. Walt, I anticipated your observation on that. So how much is that? Well, can you see the space between these two pieces of paper? Barely? That's 1 millimeter, okay? That distance is 1 millimeter. So what we're saying here is that his speed is about 3 of these millimeters in an hour. That's, okay, approximately 1/8 of an inch an hour would be the speed of one of these Kinesin molecules. Human walking is 3 miles per hour. That is, you walk at about 4,828,000,000,000 nanometers per hour. That's how fast you walk. Just to give you an idea, he's doing 800 nanometers per second, which really sounds pretty fast. And it probably is down there on the cellular scale. They're probably clipping along. But to us, up here, they're just crawling, you know. Okay. So the University of California in Berkeley, I think, did this little, I want to see how much time I got
Our
left.
brain is made of billions of nerve cells, and they're all connected. If we take a closer look, a nerve cell seems to have antennas. Most of them are receivers of information, but only one is a transmitter, called the axon. This axon is connected to several receivers of other cells, forming a gigantic neural network, the brain. Meet John. John is a kinesin, a motor protein. He lives inside a nerve cell, and he has a proper job. To ensure that a brain cell does his job properly, it needs the continuous flow of building materials, proteins. They travel through the cell using the cytoskeleton. If you would compare a nerve cell with a city, the cytoskeleton inside the cell would be the roads, and the traveling proteins would be the traffic. These materials are towed by motors along the roads. And just as in real life, there are different kinds of motors and different kinds of roads. John's sole purpose in life is to deliver his cargo to a specific place in the axon. He takes the main roads, and he walks in just one direction only. John's job may seem easy, but it's not. He has to overcome a number of obstacles to ensure that the right amount of cargo arrives at the right place. To make the journey even more difficult, John is not alone. Other motor proteins ride along with his cargo. They haven't woken up yet, but that will happen soon. The journey starts in the center of the city, just like in the center of the cell. To enter the axon, John has to pass a place called the axon initial segment. In this segment, there are 2 kinds of roads. The main roads that John uses, called the microtubules, and a lot of little alleys called actin. And here, our brave motor protein meets his first challenge, because one of his sleeping travel companions, myosin, has woken up and starts to cling to the actin. And there are a lot of alleys. Only brute force can save John now. Fate strikes again. The other companion, Dynein, wakes up, and he can only walk in the opposite direction of John, resulting in a tug of war. But there can be only one. Along the axon in which John travels, there are places called synapses. Here, the axon connects to receivers of the other cells. Regulating proteins call the shots here. This traffic police makes sure that all passing traffic gets to the right destination. If John's cargo is needed in this synapse, he will be stopped and myosins take over his load. But today, John's cargo is safe. But what he does not know, that his road is under construction, just a few blocks away. In our nerve cells, the cytoskeleton is changing constantly. Roads are built, but are also broken down. Facing this kind of obstruction, John has to find a detour. John isn't the only motor protein on the road. There are many more. Our nerve cells need a smooth traffic flow in order to perform well. A traffic jam due to problems during the journey may ultimately result in brain disease. Understanding the challenges John faces could improve treatment or prevention. Finally, John arrives at his destination. He has fulfilled his destiny. But several other Johns are just getting started.
So there you have the story of Kinesin. Interestingly, they gave him an eyeball and supposedly a brain. You understand, Kinesin doesn't have any such thing. And the questions are still out there. How does it know? What is it responding to? And I'm sure these are questions that scientists are asking and pursuing because it is a mystery. It's all part of God's creation, and we're still learning about it. Yeah.
If they would look to God, they'd know the answer.
You know, ultimately, yes. But the interesting thing is, God has given us a brain, and I think the really nice part about it is we can appreciate what God did, and we're free to pursue questions and answer them as well. And I think personally, a scientist motivated by examining a creation rather than a product of chance, an accident, is much more exciting and fulfilling along the way. Yeah.
If someone has a stroke, what goes on at that level?
Well, in that area of the brain, there's a shut-off of oxygen to, depending on how far down the line of the circulatory system where the blockage is.
Okay.
Of course, the further down the line, the more area of the brain would be affected, more blood would be shut off to a larger portion, and the damage worse.
So
A stroke or a clot in a smaller blood vessel might only destroy a small patch of brain.
the micronutrients that need to get there can't get there?
Well, no, that wouldn't affect that because what happens is the stroke is the result of a shut-off of oxygen from blood to the cell. What you've been watching here is activity within the cell itself. This kinesin is not at all affected by oxygen supply. What would be affected is the production of those ATP molecules. Remember those? Without oxygen, you can't make ATP molecules, so you're going to run into trouble. If you're not getting oxygen to tissues, I don't think I did electron transport chain here yet, did I? I did want to show you this one in my next presentation. This is not the electron transport chain, but is the other one, photosynthesis. This is the one that produces the oxygen we breathe. Respiration is the one that consumes, and that's in respiration, while you respire, to make ATP, so that you have the fuel to run the other activities of your cells. Yeah, you need the oxygen to make the ATP, not directly, indirectly. Yeah.
This process would be speeded up by a person, say, running or working or whatever in relation to, say, somebody sleeping, right?
Oh, absolutely. You certainly have more neural activity when your muscles are, when you're organizing your muscles and keeping them going. Although, you know, when I just sit and think or read, I can get kind of tired too. So the brain uses a huge quantity of oxygen in its activities, and you would think your muscles, if you're running, use a lot of oxygen. They certainly do. But the brain has a fair consumption of oxygen as well when you're thinking and involved in that kind of activity as well. Yeah, okay. So let me, I think we're all done here. Any other questions or? Ken,
We'll take questions after the break.
Okay, all right.
Just going to take a break for a couple minutes, give you some announcements. Our meetings are free, but we have expenses, so we like to pass our basket around. Thank you for your generous support. When we do ask questions, we will bring the microphone around so that you can ask your question with the microphone, that everybody can hear you. I mentioned earlier that we have the Creation Truth Conference thumb drives available. If I can keep your attention up here for a minute so you can hear what's going on, would be good. We have Creation Truth Conference thumb drives available for $49 out at the table at the end of the meeting. As MCF, we always need help. We need web help. We now have some great video help, thanks to Nate back there. We have help with all kinds of things relative to scheduling, housing, advertising, putting up posters, anything you want to do to put some more time and effort, give us a little sweat equity into MCF. We need board members. Glad to have you. Talk to Walter, Sharon, or myself after the meeting. If you want to sign up for MCF membership, it's only $20. You get a free DVD. Walt and Sharon can take care of that out at the table. If you're not on our email list, if you're new, get your name on the email list and you'll get the advertisements. I mentioned earlier, some of the folks came in later. Our presentations are now being videotaped. Last month's presentation with Doc Rock is now available on both YouTube and the Midwest Creation Fellowship website, so you can go out and see that. Sometime during the month, this presentation will be available there as well. Upcoming meetings. This is kind of a fun presentation for me tonight because I'm a protein chemist, not a proton chemist, a protein chemist. And I'll have some interesting questions and comments about some of the stuff we've been seeing, but I'm going to be the speaker next month, and I've been at least 100 hours at least into the presentation for next month, and I think you're going to enjoy it because I'm talking about information, and all the stuff you saw tonight is tied to information. My title is A Unifying Biblical and Scientific Perspective. I've got a whole new perspective on the whole issue of information and the science and the chemistry and the stuff that you've been seeing tonight, and it's exciting me personally because it's strengthened my faith in a different way that I didn't expect, and I'm going to share that with you. So make sure you're here next month. Then the month after that, and by the way, that's January 8th. We're shifting a week because of New Year's. So it will be the week after New Year's, January 8th. February 5th, our president, Walt Sievertson, in the back door back there, he's going to be our speaker for February, The Great Evolution Mystery. And he's been working on that for a couple months, I think he said, and he's excited about that. March 5th, we have a gentleman coming. Help me out with his name. Rick Magoo. The reason I ask for help is it's spelled M-C-G-O-U-G-H. Magoo. I went to Houghton College, H-O-U-G-H-T-O-N. So to me, it's Houghton. So that's why I'm not sure. So it's Rick Magoo. He was at the Creation Truth Conference in one of the breakout sessions. He's agreed to come in March. How many of you were in his breakout session? Anybody here? Good, it'll be all new. Or he could do the same breakout session. That would be good. April 9th, Carl Kirby. And then we mentioned April 7th, there's going to be a field museum training session for people to sign up for. That's in coordination with Quinton Road Bible Baptist Church. I think there'll be more information on that on our website. May 7th is Ron Dudek, going to be coming and talking about cannibalistic plants. Could be kind of fun. So with that, sign up for notifications. I don't have any other comments, so let's go to the question and answer. I just ask you that you use the microphone for questions. Helmut, you have a comment? People like to correct all kinds of things. I grew up raising Venus flytraps, and when you see them eat a fly, they're pretty cannibalistic. You can interpret that they don't eat each other. That's correct. But they have fun eating meat. So let's get some questions for Dan. Who's going to be first with the microphone? Got to get a granddaughter question in here.
Uh-oh.
Can me and Aya do more with the beads?
Can they do more with the beads?
Yeah, you can build another protein with the beads. You want to build a kinesin next. Okay, we'll build a kinesin. Kinesin.
I heard somebody mention my name over here. Okay.
If you don't get email notifications for these meetings and want them, and please print plainly because emails bounce and it's just one error.
I have announcements too. If you do get our emails, please don't bother to sign up again and again and again and again. Some people sign up every month. I promise you, I'm not throwing you off the list. If you don't get it, check your spam bin and put us in your contacts list. See if that works. And let me know. December 11th, we have a movie. The third and final showing of Genesis Paradise Lost is coming. If you go to our website, you can find where their website is to find where it's showing. January 11th, we have another movie coming, Alien Intrusion, based on the book by Gary Bates. There's something really weird going on in the world. People claiming to be kidnapped by aliens. There's something going on. And then I do have a question, Dan. What were those little knobby things hanging off of the bag that Kinesin is carrying?
Oh, those would probably be proteins of some sort, either recognition proteins or receptor proteins of some sort, perhaps channel proteins that allow objects to move in and out of that vesicle. The jobs of proteins that they do on cells and vesicles a lot. So I think the author was just trying to show you that a vesicle isn't just like a bubble and all the same, made out of the same phospholipids, but there is that membrane plus proteins stuck in it.
Is that the stuff that John is dragging against?
Well, I could admit, here we are. I need to envision this. There's a tremendous amount of resistance. You look at that animation and it looks like all space that he's walking in, and here and there's another something, inside the cell, and that is not at all what it's like. It is jam-packed in that cell. It is, there are so many different molecules at so many different sizes. It is worse than a traffic jam as I foresee it. Yes, there's water, but it doesn't create this hollow water space that things just easily kind of drift through like you in the swimming pool. You in a swimming pool with 1,000 other people jammed in the swimming pool with beach balls and floaties and everything else, and you're going from one end to the other, that would be more like the contents of a cell.
Did I notice that John was building the tubule as he was walking along? It looked to me like he was building the tubule as he was walking along.
I didn't catch it or notice that, but it
was being built just ahead of him.
Yeah, I don't know a lot about the dynamics of microtubule building, but that is how they operate. They have to. The tubule has to be in place for the kinesin to walk. Okay, and tubules do assemble and they do disassemble. And what controls that? I don't know. Maybe someone knows. I haven't heard it ever talked about. There is a plus end and a negative end. It's as far as I can tell, a mystery. And how close does it assemble in front, behind? I don't know. Yeah.
Just to comment on the motion, did anybody catch in there what made these things move? It was mentioned once in one of the videos.
Oh, you talked...
It was called Brownian motion. Anybody heard of Brownian motion? You have to realize when you're looking at these things, particularly a lot of his illustrations early that showed the colorful ribbons that were rotating, those are, while he was rotating them, they're still stagnant pictures. And you have to realize that Brownian motion is making everything in the cell move. None of this stuff is static.
No.
Everything is moving constantly. So how do they get those static pictures of those proteins? Most of them come from X-ray crystallography. Chemists try to crystallize these proteins and get them into a solid form, put them in an X-ray machine, create these incredibly complicated diffraction patterns, and put together those structures that you saw. So every one of those structures was probably a half a dozen PhD candidate theses just to put together one of those structures that he showed tonight. And then they computer animate that and have them and turn. So the level of complexity of what you have seen tonight is just absolutely unbelievable. It looks simple on the screen. It looks like it's all worked out. But realize all of this stuff is in constant motion. A few of the cells that were big enough to be seen by microscopes and electron microscopes, you could see video pictures of the actual cells. But you saw nothing other than computer animations because the stuff is so small and so complicated. And wait till we talk about information next month. Questions?
I wonder one thing. I noticed a lot of things flying here and there, the ATP coming, the ADP going out, the building blocks for the microtubules going in and out of the highway, so to speak. How are they just floating, or something is transporting them from one place to another?
Well, you remember the principle of diffusion. And molecules moving from areas of high concentration to areas of low concentration. When a tubulin, let's say when a tubulin protein connects to a microtubule and is pulled out of solution, then in that neighborhood, you'd have to imagine in just that neighborhood, the concentration of tubulin just went down. So other tubulins in solution are going to move toward that area following diffusion gradients. That's a fascinating study, is diffusion gradients. The fact, the whole principle of diffusion, why do things want to get away from other things like themselves? Put a drop of ink in water and you'll see the ink particles move away, diffuse. Why do they do that? Why do they move to a position as far from the next one as it possibly can get? And that's what it does by random movements. The cell employs that chaos to actually do work. It's amazing. Yeah, and we remember ATP synthase and the movement of protons from a concentrated area through the membrane, through that ATP synthase to an area of low concentration. Why is it doing that? It's diffusing, following diffusion gradients. Yeah, question, go ahead.
You mentioned something on one of the other questions regarding the microtubulars. You said a positive and a negative. Are we talking as in magnetism?
No.
I thought I would.
No, just, that's a good question. No, the positive and negative are just direction.
Okay.
Positive toward the periphery of the cell, negative toward the center of the cell. Yeah, Kinesin's moving in the negative direction on tubule, Dynein's moving on the positive direction. Yeah.
What is it carrying towards the inside?
Well, it was inferred in that one funny one that the Dynein's bringing Kinesin's back. Did you see him? That one Dynein molecule was kind of strutting along and dangling from behind him was a Kinesin. Well, you know, well, wait a minute, he's moving to the periphery, Dynein would be, so he wouldn't be carrying Kinesin that way. That would have been the other way around. I don't know what moves them in which direction. I mean,
how many different kinds of cargoes do these guys carry?
Well, like in that case was a vesicle that contained neurotransmitters. It could be vesicles containing hormones. Could be vesicles containing, well, there's all kinds of protein products that need to be moved within the cell. Even organelles themselves, moving mitochondria and evenly distributing them in the cytoplasm so that they're more efficient in their production of ATP, and then how ATPs get to other reactions more efficiently, moving them, spacing them out.
So that's the undifferentiated protoplasm that they were talking about 100 years ago, huh?
Yeah, I guess so. It's much more complicated now as they've finding all these proteins in there. Yeah.
Realize, folks, this is groundbreaking research. It's all new. We don't have all the answers yet.
Oh, yeah.
Kinesin, the molecule kinesin was named John in those last video clips.
and it goes from the inner to the outer edge of the cell. When it gets to its destination, does it disintegrate or does it stay together and maybe dynein carries it back to the inner of the cell for the next trip?
Yeah, it could be. It wouldn't be unreasonable.
Or does it get disintegrated and then new ones form in the inside of the cell?
Yeah, that's probably the most likely. Proteins wear out. They do wear out, and they're recycled by the cell. They spontaneously denature and come apart. So when they're first constructed by the ribosome, they're in their most stable form. Now, as they do their work, here a coil pops off, and here a fold comes unfolded, and eventually that protein is spontaneously denaturing and is useless. There are other proteins who gobble them up. There are even digestive vesicles which will engulf those, break them down into amino acids, and distribute those into the cytoplasm, where they can be recycled and rebuilt under the instructions of DNA into fresh proteins. Proteins have a certain life expectancy. They don't last. Well, I can't say how long. It will probably depend on the protein. But that's not unreasonable to think. They probably just wither away and are recycled.
One of your last cartoons showed a withered kinesin being carried back by the Dyson.
Okay, yeah. Proteins do wear out, as we all do.
You can see room for a couple typos here and there in the genetic code.
Yeah.
What causes a cell to die? And when it does, what happens to everything in it?
I once showed a slide which represented the biochemistry that takes place in a cell. I don't know if any of you recall that slide, but it was this extremely complicated collection of biochemical reactions, all of which are taking place in the cell. When they're all functioning properly, we say the cell is alive. If that chemistry at any point shuts down in some location, begins to shut down, if it's vital chemistry, of course, the cell will stop all chemistry and we'll say it's now unalive, and the word for that is dead. Chemistry can shut down gradually. There are less vital reactions than other reactions, and so if a less vital reaction shuts down, the cell is weakened, but it may still be alive. That can happen as well. But it is the gradual loss of the overall metabolism of a cell, which when a certain critical level is reached, if that chemistry is not maintained, the cell will simply die. You understand that you turn over cells, your entire cell mass as you're sitting in that chair right now, 7 years ago did not exist. Every cell in your body, except neurons now, every cell in your body has been turned over. Within 7 years, every cell in your... So, the person you were, the cell collection you were 7 years ago no longer exists. But because of mitosis and cell division, new cells are produced to replace the cells and let them die off the other ones, and newer cells, fresher cells with fresher proteins, keeps you going. Yeah, about every 7 years you've completely turned over all your cells. What?
It's this process thermal related. In other words, as it's cold, is it accelerated by increased temperature?
Yes, oh, absolutely. The question was, does this chemistry change with temperature, increase with... Yes, it would. It naturally would. Cellular kinetics always follows the pattern that a rise in temperature would increase molecular movement, atomic movement, and that, of course, would increase cell reaction tempo, and so forth, yes.
So
and you
this would also be affected by hibernation of certain species?
Yeah, under hibernation conditions, torpor and hibernation where animals will, they don't choose to, but they can selectively shut down or slow down system tempos, whether it's blood pumping tempo, hormone production tempo. Yes, this can happen in certain animals to where those are slowed to extremely low rates, sometimes because of temperature, sometimes because of food supply that's unavailable or water is unavailable, and the need for the animal in order to survive to simply shut down, slow down certain reactions, less vital reactions, of course. Respiration being the one you can't ever shut down entirely. You can slow it down too, but there are, and again, we talk communication and controls. How this is all regulated and controlled, I have no idea. And for you students, there is so much work that could still be done. There are innumerable PhDs out there that can be gotten if you'll jump into one of these places and do the research. I mean, there's a lot we don't know. Question back there?
I had just a comment, because I've never come to this before, and it's very complicated, so I'm not a scientist. So now I want you just to pull this back so it might entice me to come again, because it was over my head. But how, because I know Dan is a Christian, and I want him to just tell how this fits into creation. I think part of what I heard him say was, how could the complexity of our bodies not come from just something that's more than just a big bomb theory or something crazy like that?
Right. You know, I know Joan is a nurse, a registered nurse, and her husband, Ben, there is an engineer. When we look at this, and you looked at it, you just saw a little bit of it, we can't help but see, design was mentioned, but there is engineering here. We're dealing with materials, in our engineering world, we may deal with steel and concrete and glass and plastics, yes. In God's world, it's proteins. Basically, proteins is what he's working with, but the engineering is still there. I'm still wondering, did this happen like that in the mind of God? Or was there a period of time where he sat down with angels and said, hey, let's engineer and call it life. Let's make atoms and okay, I know that's silly. But when we look at what's going on, we can't help but see design and purpose, planning, even recovery methods, healing, recycling. It's all in there. It's all in there. That doesn't just happen. Evolution wants to tell us, at least on its raw core premise, is that this is all the product of chance. Well, I've watched chance operate. It creates chaos. It doesn't create order. It doesn't create engineering. No, whatever has done this was a mind. Amen. There's a mind behind all of this. I'm sorry, there is no chance for evolution. Don't even tell me it's 1 billion to 1 odds. I won't even give you that. There is 0 chance that this can happen by chance. There's a mind behind this. There's a mind behind anything we see that's engineered. So.
Please come next month. We're going to unify it next month. Any other questions?
So when the gentleman up here was talking about temperature speeding up the processes, are you talking about environmental temperature or body temperature?
Well, okay, both, in fact, because even as you sit here right now, the temperature of this room is affecting you, plus your biochemistry's at a rate that you're generating 98.6 degrees worth of heat energy off your body, and the difference in the two is either causing you to sweat right now or you're cool. Okay, so the temperature we're talking about in a cell is temperature mostly due to the metabolism within the cell. That is, the chemistry going on in the cell is generating heat. The cell has a way of controlling that. Water is one of the biggest controls. The cell is mostly water, and water has a very high specific heat. It can carry off a lot of heat, and it uses that. Where does it go with it? Well, it takes it to your skin's surface, and you radiate off that heat that way, so that you can control how much you radiate off by how much you sweat, so that your body maintains the 98.6, plus or minus 0.5, what have you, in that range, always. Now that temperature does affect the chemistry. but the chemistry of your cells is used to 98.6, and it's comfortable and runs smooth at 98.6 degrees. If you begin to drop too low, you got problems. If it begins to go too high, you know you got problems, okay? Because the chemistry is used to 98.6 for you, okay? Now that's not the same for all creatures. Temperatures are different.
So is that why we feel so tired when we have a fever, because everything is sped up and?
Well, you've called into motion your immune system. If you've got an infection, whether it's viral or bacterial or even parasitic or fungal, whatever the infection is, your body's under attack, and you have a defense system for that. You have an immune system. It's always operating. But when it does detect a problem, it goes into hyperdrive. Additional antibodies are produced, white blood cells are produced, and all of this defense mechanism now goes into drive to reduce that infection. So that's going to take more chemistry. That's going to drive the metabolism of your body up, probably. You'll have a temperature spike, an increase, and so much is allowed. You know that. But if it gets to what, 104, Joan, help me out here. Time to get to the emergency room. That's a little too much. But you've called extra resources into play, and that's going to increase temperature. It's going to consume resources. That means you're going to be hungry, probably, afterwards, because you've called into play this additional system, the immune system, under infections.
And as you said earlier, all of this requires energy, so you're using up more of your ATP than you would normally.
Yep.
So your system feels tired. You don't have enough available
You will be tired.
energy.
Yeah. Other things will slow down, shut down even.
8:28, any more questions? Going once? Going twice? Sold. Let's thank Dan.